Today we're going to do the notes on 5.4, which are all about heredity. So how do parents pass on their traits to their offspring? Oops. Okay, so first, like I just said, heredity is just the process of passing on traits from parents to offspring. I'm not going to play this video for you in the notes. It is linked in your notes document, so if you would like to watch it, please click on this link. It's a good overview of just what DNA chromosomes are and like the role they play in passing genes from parent to offspring. But that's basically what heredity is the process of parents giving their traits to their offspring. We can't talk about heredity without talking about the scientist Gregor Mendel. He was a monk in Austria, and he was a gardener. He did most of his studies with most of his recorded studies were in the garden with pea plants. Okay, so he took very, very meticulous notes, and he crossbred and had a very controlled crossbreeding system where he would cross pollinate using probably like paintbrushes, but he would cross pollinate certain plants and see what were the outcomes. You know, what kind of offspring plants do I get if I cross pollinate this plant with this plant? Okay, so some of the characteristics that he looked at were things like the color of the seeds or the color of the actual peas in the plant or in the pods. So the seed color, yellow or green, the seed shape, some peas are round, some peas are wrinkled, pod color, green or yellow, flower position, flower color. Sometimes you'll see, a lot of times you'll see example problems involving pea plants, and all of that is because of Gregor Mendel studies. Usually you'll see those studies or those example problems involving like the color of either the flowers or the peas themselves, or maybe even the shape of the peas themselves, if they're round or wrinkled. That's typically the type of heredity problem that you'll see. This is a video that just talks about Mendel and his pea plant studies.
These days, scientists know how you inherit characteristics from your parents. They're able to calculate probabilities of having a specific trait or getting a genetic disease according to the information they have from the parents and the family history. But how is this possible? To understand how traits pass from one living being to its descendants, we need to go back in time to the 19th century and a man named Gregor Mendel. Mendel was an Austrian monk and biologist who loved to work with plants. By breeding the pea plants he was growing in the monastery's garden, he discovered the principles that rule heredity. In one of the most classic examples, Mendel combined a purebred yellow-seeded plant with a purebred green-seeded plant, and he got only yellow seeds. He called the yellow color trait the dominant one, because it was expressed in all the new seeds. Then he let the new yellow-seeded hybrid plants self-fertilize, and in this second generation he got both yellow and green seeds, which meant that the green trait had been hidden by the dominant yellow. He called this hidden trait the recessive trait. From those results, Mendel inferred that each trait depends on a pair of factors, one of them coming from the mother and the other from the father. Now we know that these factors are called alleles and represent the different variations of a gene. Depending on which type of allele Mendel found in each seed, we can have what we call a homozygous pea, where both alleles are identical, and what we call a heterozygous pea, when the two alleles are different. This combination of alleles is known as genotype, and its result, being yellow or green, is called phenotype. To clearly visualize how alleles are distributed amongst descendants, we can use a diagram called the Punnett square. You just place the different alleles on both axes, and then you figure out the possible combinations. Let's look at Mendel's peas, for example. Let's write the dominant yellow allele as an uppercase Y, and the recessive green allele as a lowercase y. The uppercase Y always overpowers its lowercase friend, so the only time you'd get green babies is if you have two lowercase y's. In Mendel's first generation, the yellow homozygous pea mom will give each pea kid a yellow dominant allele, and the green homozygous pea dad will give a green recessive allele, so all the pea kids will be yellow heterozygous. Then, in the second generation, where the two heterozygous kids marry, their babies could have any of the three possible genotypes, showing the two possible phenotypes in a 3-to-1 proportion. But even peas have a lot of characteristics. For example, besides being yellow or green, peas may be round or wrinkled. So we could have all these possible combinations: round yellow peas, round green peas, wrinkled yellow peas, and wrinkled green peas. To calculate the proportions for each genotype and phenotype, we can use a Punnett square too. Of course, this will make it a little more complex. And lots of things are more complicated than peas, like, say, people. These days, scientists know a lot more about genetics and heredity, and there are many other ways in which some characteristics are inherited, but it all started with Mendel and his peas.
So they went into a little bit more in depth than we need to about crossing more than one trait. Most of our examples, we're only going to be figuring out one trait. But when Mendel was studying his plants, he noticed there are some plants that are what we call true breeding. This is what today we might call like a purebred or a homozygous dominant or homozygous recessive plant or organism. So he found out that some plants were true breeding for a specific color, either green or yellow. So then what he did was he crossed a true breeding or a purebred green pea plant with a purebred yellow pea plant, and he noticed that in his first generation, all of the offspring were green. That yellow trait seemed to completely disappear. So that's what she was just talking about in that video, about some of those traits can be hidden. This is what he described as that trait kind of receding. Okay, this is what we call a recessive trait. So if you were to cross a true breeding green plant and a true breeding yellow plant, cross-pollinate them, you end up with all green plants. This tells us that that green trait is dominant. That means like if that offspring plant even has just one trait from one parent, that green trait is going to take over, and that's what's going to show. That yellow trait still gets passed on to the offspring, but it's kind of hidden. And the reason I know it still got passed on is because Mendel then did a second generation cross. So he took two of those green plants from that middle generation and he cross-pollinated those plants. When he cross-pollinated those plants, he noticed my offspring generally came out green, but sometimes I did see that yellow trait pop back up. So he crossed some of those first generation plants with each other. He noticed that 3 out of 4 offspring plants probability was green, 1 out of 4 was yellow. So this study led us to now more of our modern genetics probabilities, which then gave us the ability to predict genetic crosses using Punnett squares, which is what in that last video they showed us talking about those little boxes that they were filling in with the Y's and the R's. And we'll talk about in this lesson, we'll talk about how to complete those Punnett squares.
Stated Clearly presents, what exactly is a gene? Each one of our cells contains 46 strands of DNA. A single strand is made of millions of particles called nucleotides, and these nucleotides come in 4 different types, which scientists have labeled A, C, T, and G. A gene is a special stretch of DNA, a sequence of A's, C's, T's, and G's that code for something. A gene contains information for a cell to read and use, but what exactly does that information do? You might have heard that there's a blue-eyed gene, a freckle gene, possibly even an anger gene, but single genes don't literally make things like eyeballs or freckles or temper tantrums. Genes make proteins. Those proteins then interact with each other and all sorts of chemicals inside the body to build things like eye pigments, freckles, and mood-altering hormones. A single strand of DNA contains thousands of genes or unique protein recipes. Humans have roughly 20,000 altogether. Some genes are small, only about 300 letters long. Others are well over 1,000,000. The length and sequence of a gene determine the size and shape of the protein it builds. The size and shape of the protein determine the function that protein will have inside the body. Hemoglobin, for example, is a protein structure found in red blood cells. Its unique shape and size allow it to capture oxygen molecules when blood flows near the lungs, and then release them later when blood flows near oxygen-starved tissues. Pepsin is a digestive protein. Its unique shape allows it to break down food inside your stomach so it can be absorbed by the body. Keratin is a structural protein. Its unique shape and size allow it to link together with other keratin proteins to form hard structures like fingernails, claws, and beaks. Different creatures have different genes, which is ultimately why their bodies look and function differently. But one of the many reasons scientists believe all life on Earth is related is that the basic DNA code, the language of A's, C's, T's, and G's, is pretty much the same for all living things. Many creatures even share some of the same genes. You might not be too surprised to learn that humans and chimps, which are closely related, share 96% of their genetic code. But what would you think a lowly fruit fly has in common with a beautiful swimsuit model? Surprisingly, about half of its genes. Because all creatures use DNA in pretty much the same way, genetic engineers have found that if they take a gene from, say, a bacteria cell and insert it into the DNA of an animal or a plant cell, that animal or plant cell will then read the new gene and produce the bacterial protein. Engineers have mixed and matched the genes of different organisms to produce many new creatures, including corn that is toxic to insects but supposedly safe for human consumption, tomatoes that last up to twice as long in the grocery store before going bad, and a new form of bacteria that produce the human protein insulin, which we then collect from these bacteria and give to people with diabetes who need extra insulin to survive. So just to sum things up a bit, what exactly is a gene? A gene is a special stretch of DNA, not the entire strand of DNA, just a segment that codes for something. Each gene is like a unique recipe, which usually tells a cell how to make a protein or a group of proteins. Different creatures have different genes, but all genes are written in the same basic DNA language of A's, C's, T's, and G's. I'm John Perry, and that's genes stated clearly.
Okay, so how are those genes passed down from parent to offspring? When those gametes combine, that is going to give the offspring a set of genes from biological mom and biological dad, or from one parent and another parent. There's different versions of the genes. Some genes can have dominant versions of them. Some genes have recessive versions of them. Some genes have more than one dominant versions of them, that like eye color, that kind of thing, and more than one recessive version. But all the different versions of a gene is called an allele. So this word here, allele, is pronounced allele. We typically, in 8th grade science, will just deal with genes that have one dominant and one recessive. There's a couple of exceptions, but we'll typically just deal with one dominant and one recessive trait per gene. Dominant alleles are written with capital letters. Recessive alleles are written with lowercase letters. So to kind of give us that example that we used a couple of slides ago with those green and yellow pea pods, the green would be a capital, probably G, and then the yellow would be a lowercase g. You would not end up using different letters because we're talking about the information for the same gene. You're going to use the same letter, just one's capital, one's lowercase. If an organism has two of the same letters, then it's what we call homozygous. Okay, homozygous. Homozygous just means that they have two of the same letters. So an organism can be homozygous dominant for a trait, meaning it has capital that it got from both parents, or it could be homozygous recessive, meaning it has lowercase that it got from both parents. One's not better than the other. Dominant doesn't necessarily mean better. It's just this is the trait that takes over if it shows up. If an organism has one capital and one lowercase, or one dominant and one recessive from one parent or the other, then that's what we call heterozygous. That prefix hetero means different. That prefix homo means same. Prefix hetero means different. So if an organism is homozygous dominant for brown eyes, then you would represent its traits with capital B, capital B. If an organism is homozygous recessive for, let's say, red hair, you would maybe represent it with like a lowercase r, that kind of thing. If the organism has brown eyes, but they carry a recessive blue-eyed gene, then maybe you would have a capital B, lowercase b. That's heterozygous. Okay, so they have brown eyes, but they carry that blue-eyed gene that they could pass on to their offspring. Okay, so the combination of alleles that you have is called your genotype. So if you have capital lowercase, capital, or lowercase, depending on what genes you have, that's called your genotype. What you see or what you can observe is called the phenotype. So that example I was just giving about, I could have a capital B, lowercase b genotype for my eye color, being I carry a brown-eyed gene and a blue-eyed gene. So that's my genotype. But what you see is just brown eyes. And what you see in my phenotype isn't going to look any different from if I were to have capital dominant. Dominant genes will show in your phenotype, even if you only have one. Recessive phenotype will need to show, recessive alleles, you'll need to have both recessive alleles from both parents in order for that to show up in your phenotype. So even if you only have one dominant letter for a gene, that's what's typically going to show up in your phenotype. And we'll see several examples of this as we do some practice Punnett squares. So I know this is a little bit, it sounds a little bit complicated with all of these terms: homozygous, heterozygous, phenotype, genotype, zygote, all that stuff. I know it's really a lot of information, but we'll get some practice with our practice problems today. Sometimes we have situations where lots and lots of genes put together can influence one trait, and eye color is one of them. Typically, eye color, hair color, skin color, things like that aren't so simple as just saying, oh, I've got a capital B, so I have brown eyes, and I've got a lowercase b, so I've got blue eyes. It's not so simple. So typically, in humans, especially because we're such complex organisms, a lot of genes can influence one trait. Sometimes one gene can influence lots of traits. This is the case for sickle cell anemia. Sickle cell anemia is what you see in this bottom right-hand picture here, this diagram. This is a condition where instead of blood cells being kind of like inner tube or donut shaped, even though there's not a hole in the middle, it's more of an indent. But instead of that donut shaped blood cell, some of the blood cells are shaped like a sickle, like a crescent moon kind of thing. This can happen, or this can cause a lot of issues with low oxygen levels and veins and arteries getting blocked, and issues with circulation and blood flow. But typically low oxygen levels is a really big problem for people with sickle cell anemia. Okay. Sometimes the environment can influence traits or can influence what you see. A really good example of this is, I really love this picture here of the Arctic fox. The Arctic fox is just one of tons and tons of examples of an animal that has the environment having an impact on its phenotype. So the environment can influence what traits it has. In the spring summertime, the Arctic fox is like more of a brownish tannish color to blend in with the environment. And then in the wintertime, you'll see the Arctic fox's fur turn completely white. And we have some Arctic foxes at the Columbus Zoo, and in the different seasons, their fur turns those different colors. It's very, very cool. Another example is human skin. So like the melanin in our skin has our skin getting a little bit darker if we're exposed to UV light for long periods of time. And this is just an example of how the environment can influence our traits. That's an adaptation that our skin does to kind of help protect us from those UV rays. And this Arctic fox, that's an adaptation that Arctic fox has to help protect it to hide so it can hunt a little bit easier, so it can hide from its prey as it's trying to stalk, and also probably hide from predators as well. There are a couple of exceptions here. So everything we've talked about so far is what we call complete dominance, where a trait is either dominant or it's recessive, okay? There are some exceptions to this. One example is what we call incomplete dominance. We're typically going to see examples of incomplete dominance using flowers. So an example of incomplete dominance would be like if I were to take a red flower and a white flower and cross-pollinate them. You're not going to end up with red or white or some red, some white. You're going to end up with a blend of the two, okay? So for heterozygous individuals that have both traits, you'll see a blend of the two. A blend of red and white is pink. So this slide is showing an example of incomplete dominance between a red flower and a white flower producing pink offspring. There is something that I would like to address here on this slide. This lowercase r for my white flower, I disagree with that. Typically, for an incomplete dominance situation, you would see another capital letter, but it would just be a different letter. So maybe it would be a WW. And then each one of these offspring would have an RW, capital. This is because red is not any more dominant than that white. They're equally dominant. They blend with each other. Okay, so they're just, neither of them are completely dominant over the other one. Sometimes you'll see it written as like a little superscript, meaning like the letter that shows the color is like tiny and high up, like an exponent. Sometimes you'll see it written that way as well. Another example of an exception to those complete dominant situations is called codominance. This is where you don't have one color dominant over the other completely, but what you see on an offspring that has both traits is not a blend of the two, but you'll see both dominant traits kind of showing up in spots or stripes. Okay. A really good example of this is in horses and cows. And this is what we call the roan coat. So this is what we see on the right side of this slide here. You cross a red, which is really more of a brownish color, but they call that coat color red. You cross a red cow with a white cow. They're both equally dominant. One's not going to take over the other. And you end up with the roan coat. So on that offspring that has both of those traits, you'll see parts of that coat are red, parts of that coat are white. Okay, so that's what we call codominance. That prefix co means like with or together. So instead of it being incomplete where you see a blend where it would be like a light brown or a tan color, here you're seeing both of those dominant traits. Neither one is giving way to the other. They're not blending together, but you're seeing them both work together in a codominant situation. This image I really like. This one this shows like how a red flower and a white flower could cross in two different ways. If it's incomplete dominance, over here on the left, you're going to see red with white making a pink flower. If it's a co-dominant situation, you'll see red with white making a flower that has red and white in it. This is from page 333 in your textbook, and this just wants us to draw exactly what we just did on that last slide with that flower. But if I were to have a blue fish and a yellow fish, and they were to exchange genes to pass on to offspring, what would their offspring look like in each one of these situations? In a complete dominant situation, if blue is dominant to yellow, according to this picture here, then my offspring would be a blue fish, okay? If it's incomplete dominance, then you're going to see those colors, the blue and the yellow, blend together, and you're going to see a fish that's green, okay? Blue and yellow make green, if you remember from elementary school. In a co-dominant situation, if I cross a blue and a yellow fish, you're not going to see one giving way to the other. You're not going to see them blending, but you're going to see both blue and yellow in that offspring. So you're going to see a fish that has blue and yellow.
What do snapdragons, human height, and speckled chickens have in common? Well, they are easily identified as non-Mendelian traits. By non-Mendelian, we mean that, genetically, they are rule breakers. They don't follow the regular Mendelian rule that having a dominant allele means the dominant trait will show. Remember in our guinea pig video, having a dominant allele represented by a capital H meant that the guinea pig would have hair? Only if there was no dominant present, a genotype of little h, would there be a hairless guinea pig. Well, that's a Mendelian trait, but what we're going to cover now is when these traits are non-Mendelian and they don't follow these basic rules. So let's first start by snapdragons. We confess that when we heard this word, we thought they were some really amazing kind of creature. Well, they are amazing, but they're flowers, so I don't know, that's not exactly what we envisioned. Well, in snapdragon genetics, there can be 3 phenotypes: red, white, or something in between, pink. It's called incomplete dominance. In incomplete dominance, the dominant allele is not completely expressed when the recessive allele is around. There isn't really a clear dominant allele. If you cross a red flower, and we're going to write it here big R, and a white flower, little r, you are going to get offspring that are big R, little r. But unlike a Mendelian trait, if this is incomplete dominance, that big R allele is not completely expressed when the little r is around. So big R, little r in this case is pink. If you cross 2 pink flowers, big R, little r, like shown in this Punnett square here, you can get offspring that are red, white, or pink. Incomplete dominance is different from co-dominance. Co-dominance, like a co-worker, that prefix co should make you think together. They work together. The alleles, that is. For that reason, we like to use different letters entirely to represent the alleles. There is a codominance involving color in some types of chickens. Take a look at this Punnett square. If you cross a black chicken, represented here by BB, and a white chicken, represented here by WW, all the offspring here are BW. BW chickens are both black and white. They're speckled. See, both traits show up. This is the essence of codominance. And you know, what's more awesome than a speckled chicken? Well, except for those silky bantam chickens. By the way, you will find that many people use different formatting for incomplete dominance and codominance. Some people like to use different letters entirely for incomplete dominance, and some people don't. Some people also prefer to do exponents with codominance, and some people don't. Formatting aside, when you're solving these Punnett squares, since we find this really does vary from classroom to classroom, the real concept is that in incomplete dominance, one allele is not completely dominant over the other, so you see this almost in-between phenotype. In codominance, both alleles are expressed. It's the application that matters here. Height is fascinating. In our immediate family, Pinky is taller than Petunia. Our mom is also taller than Petunia. So how does that happen?
there isn't just one height gene. There's a lot of genes that determine your height. What I mean by that is that you don't just have a pair of alleles, like big A, that will code for your height. It's more like someone having a genotype of big A, big B, big C, little c, and maybe more genes to ultimately determine your height. And you inherit one allele for each of the height genes from each parent. All of these genes work together to determine your height. Your skin color is also determined by many genes, just like your height. These are called polygenic traits. Poly means many, so many genes coding for one trait is what polygenic means. By the way, both height and skin color can be influenced by environmental factors as well. Nutrition growing up can affect your height, just as spending a lot of time in the sun can affect your skin color. However, this doesn't change the genetics for this trait. And finally, one more thing that we want to talk about. Epistasis. Epistasis is when one gene really depends on another gene for it to even be expressed. I mean, it really depends on this other gene. So let's pick an animal like, a llama. Let's say this llama has a dominant big B allele, which means its wool will be black. So big B, or big B, little b means that it will have black wool. And let's say that if a llama has a pair of recessive alleles, little b, then it will have brown wool. Now, what if there is another gene that controls whether this pigment will even be expressed in the llama in the first place? In this hypothetical example, a llama could have this other gene coded for by big C, big C, or little c in its genotype. However, if a llama has the genotype little c, it will not allow the other gene for wool color to even be expressed. Since we have two genes here, the gene for wool color and then this gene that controls whether the color is even expressed, this calls for our favorite 16-square dihybrid cross. If you notice in this dihybrid, crossing two heterozygote llamas, any llama with big B and big B will typically give a black llama, and little b will typically give a brown llama. In all cases, unless they inherit a little c. If the llama has a little c in its genotype, then the gene for wool color is not expressed, and the llama is albino. This means that no pigment is expressed at all. Pretty interesting. There are many other non-Mendelian traits. We have another video on multiple alleles and a video on sex-linked traits. These are also non-Mendelian. It's kind of fascinating to see what can happen when you have these rule breakers. One last thing. Keep in mind that when problem solving in genetics, you do not want to just assume it's non-Mendelian unless you are provided information or clues in the problem that it might be. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.